Hi everybody, this is Dr. Dan and I'm back with another segment of our lab. So now we have been growing out our cells on our 96 well plates for our cytotoxicity assay. It's time to add our chemicals. So we're going to add the chemicals for 24 hours and then do the neutral red assay tomorrow. So I've already started getting things prepared. You can see I got things in the biological safety cabinet. I have all our chemicals in the water bath over here that we made last time. So I'm going to get these out, spray them down, put them into the biological safety cabinet. Okay, so I put all the chemicals and media in the cabinet, and remember I'm doing two different plates rather than you guys would have only normally have to do one single plate. But I did this so that we could have four different basically experimental conditions. I have ethanol and glucose in plate 1, in plate 2 I'm testing antibiotics and bleach. Okay, so the first thing I'm going to do is I actually want to check on these cells to make sure they're okay. The problem is when there's that media in there, it's really hard to see the cells at the bottom with the microscope. It's even hard with the fog at the top here, but I'm going to take off the media first and just take a real quick glance at cells so that we can make sure they are in good shape before we start adding chemicals. Because if we're adding chemicals to places there's not cells, that's not a good thing. Okay, so I'm going to take a look at these in the microscope. Ideally, I'd take the lid off, but that would definitely contaminate the cells, so we're going to keep the lid on.
This is a picture of the cells in the microscope. They look very good in all the wells. You can see there is a ton of cells.
Okay, so these cells look really good. There is a ton of cells in there. Maybe even too many, but that's okay. We're going to go ahead and proceed. So I'm going to go ahead and add all the experiment groups in here. I'm going to start off with the controls. So remember that we need about 180 microliters of each solution. I made about 1 ml of each, but if I put 180 microliters in there, then that will be fine. So I didn't make enough of this control DMEM only and bleach. I just noticed that for both of these. That's okay. I'll make some more when I do the second one. Okay, and so this is where we have to be really careful, right? This is where it's easy to contaminate things. So I'm going to make sure I only have one thing open at a time. Open this. Okay, so that one's done. Now move on to the bleach. Alright, so I have my controls now. I'm going to speed through the rest of these. I'll show you guys. One thing I'm going to say, it takes a lot of concentration, right? To make sure you get everything in the exact right wells. We're going to do this 60 times. I'm going to do it 120 times. It's easy to lose focus doing stuff like this. And just, you start messing things up, and it's going to affect all your results. So it's really important to maintain like this, just a very focused mindset while you're doing this. All right, so I'm going to put, and even, right, remembering that I want to put the ethanol cases vertically, not horizontally, right? You have to think about what you're doing at every step, or else you'll make a mistake. And that's where it's good to have a lab partner too to help you out, because they are very useful in reminding you of what you need to do. So I'm starting with my highest concentration of ethanol first, which I'm putting in column B over here. And you see what I did? I even skipped one, right? Like, and so I need to go back and put it in that one. So like I said, there is definitely a challenge of concentration. Okay, so I finally added all the chemicals to this whole plate, and I don't think I screwed up any. It does take concentration to make sure you focus. And so I'm going to go put this in the incubator, and then I'm going to go ahead and do the other plate. I don't think I need to record video of me doing that. It's just the exact same as I just did. And so then I have one more thing to show you, which was making the neutral red solution, and so I'll do that after I do this other plate.